Healthcare bioconvergence market size, share, and trends. The global healthcare bioconvergence market size accounted for $140.85 billion in 2024. Healthcare bioconvergence market size, share, and trends, 2024 to 2034. So the market share for the bioconvergence was 140.85 billion in 2024, grew to 152 billion in 2025, and is projected to surpass around 302 billion by 2034, representing a healthy CAGR score of 7.94%, and the category is healthcare. Okay. Let's see. Here's a graph for you. Healthcare bioconvergence market key takeaways. Europe generated more than 30% of revenue share in 2023. The drug discovery application segment dominated the market and captured more than 21% of revenue share in 2023. What's this? The regenerative medicine application segment is expected to expand at the fastest CAGR of 9%. What's that? What is regenerative medicine? Center just fallen asleep again. Dr. Michael Levin and his problem with putting frog eyeballs where they belong. Regenerative medicine is a field that uses science and technology to repair or replace damaged cells, tissues, or organs. The goal is to restore normal function and treat or cure diseases. Tissue engineering, personalized organs. Regenerative medicine could allow patients to have personalized organs made from their own cells, which could eliminate the risk of infection. Gene therapy. Regenerative medicine uses cells, molecular communication, as delivery vehicles for therapeutic agents, such as genes and cytokines. Potential benefits. Regenerative medicine could help cure conditions that are currently difficult to treat. Regenerative medicine could help restore function lost due to aging, disease, damage, or defects. Challenges. Developing regenerative medicine technologies can be challenging, and there are challenges with ensuring the safety and effectiveness of regenerative products. Regenerative medicine seeks to replace tissues or organs. What is an example? Examples include cell therapies, the injection of stem cells or progenitor cells, immunomodulation therapy, regeneration by biologically active molecules administered alone or as secretions by infused cells, and tissue engineering, transplantation of laboratory-grown organs and tissues. It's amazing, isn't it? What you can find when you look up words in context. Woohoo! Regenerative medicine, tissue engineering, biomaterials, and stem cell therapy. Yay! Isn't this great? Logging in with your phenotype, and now that they have a biocyber interface, they're going to remove some tissue, maybe mix it up in vitro in the lab, grow you a new organ, and put it in you. I'm not kidding. Cells and tissues, matrices and scaffolds, biologics, enabling technologies. Apologies. There we go. Rehabilitation, external stimulation, therapeutic physical activity, assistive technologies. Optimize clinical and patient outcomes, enhance tissue organ function, reduce disabilities, and increase the quality of life. Isn't it great? It's wonderful. Hang on, I'll get you the other one. Stem cell therapy, RNA therapy, transplantation, scaffolds, gene therapy, and our recent favorite, organoids. What is regenerative medicine? A branch of medicine. Medicine. Vice at Harvard and Vermont University. They got lots of them. They uses tissue engineering and molecular biology as a way of replacing, engineering, and regenerating human cells, tissues, and organs. Cellular therapeutics, extracellular vesicles, anti-inflammatory immune cells, cellular patches, challenges and hurdles, clinical trials, advancement, and goals. Okay? They want to avoid whole organ transplant, tissue engineered constructs. Here's another one. And we've got stem cells, synthetic niche. There's that viscoelasticity. See, it says F up here. Yeah. Yeah, when we're running the Internet of Bio-Nano things, pressure is a pretty big deal in the veins and arteries. Let's see where we are in the Mooney Laboratory. Oh, we're at Harvard. Yep. There is a tremendous need for new strategies to promote the regeneration of musculoskeletal, skin, vascular, and dental, oral, craniofacial tissues due to the large number of patients suffering from disease or trauma to these tissues. So let's get busy. Biomaterial-assisted cell delivery. Tissue regeneration can be improved by using biomaterials to exploit cell biology. Biomaterial-assisted cell delivery. To address the current limitations of cell transplantation and rebuilding tissues, e.g. large-scale cell death, we have been designing biomaterial carriers that maintain cell viability during transplantation while subsequently stabilizing the cells or mobilizing to disperse and engraft through a large tissue volume in vivo. Additionally, we are exploiting our expertise in drug delivery to create new networks of blood vessels within regenerating tissues and reestablish innervation as capillary networks are built. B-U-I-L-T. Figure 2, biomaterial-assisted cell delivery. Cells harvested for therapeutic delivery can be expanded ex vivo and modified as needed before being precisely delivered using biomaterials. I said wireless tissue engineering. People kind of tuned me out. They weren't ready to head to Harvard. And trust me when I tell you, if you watch enough of Dr. Michael Levin, the main thing he talks about is your bioelectricity and how your bioelectricity works with the morphogenic fields of your lymphatic system to heal your body. Well, they manipulate that so that when the frog was healing, the eyeballs went on its tail instead of its head, and the same thing with the plant worms and all that stuff, because they figured out that bioelectricity and telling your cells what to do will change their anatomical outcome. Thus, instead of the eyeballs going where they would go, they're reprogrammed to grow on the tail, or they're reprogrammed to grow on the hands, or whatever. Yeah, that would be your bioelectricity. No, I am not joking. Cracking the bioelectric code, again, for those of you that haven't been here. 2013, Dr. Michael Levin, probing endogenous ionic controls of pattern formation. Because your bioelectric cues function alongside chemical gradients, transcriptional networks, and haptic tensile cues as part of the morphogenetic field that orchestrates individual cell behavior into large-scale anatomical pattern formation. Where's your bioelectricity? You don't have any, remember? Right. So, that is what regenerative medicine is. We talk about it here a lot, but you guys don't. Try to tell you this is life extension technology. Okay? And just, once again, as always, want to make sure that we hit those Agenda 2030 goals so people understand that we're not talking about something sitting in Harvard that they don't already have plans for or uses. Human augmentation is a big deal. Yeah, it's got market share as well. And part of it is that same wireless tissue engineering for disease-free bodies, enhanced intelligence, optimized diet, extreme aging. Okay? Human augmentation. I mean, just have fun with that. So, next. Harnessing the power of AI for human living organoid research, 2024. And what are we in here? National Center for Biotechnology Information. As a powerful paradigm, artificial intelligence is rapidly impacting every aspect of our day-to-day and scientific research, day-to-day life, and scientific research through interdisciplinary transformations. Living human organoids have a great potential in vitro laboratory, reshaping many aspects of in vivo true human organs. In vivo is in place of origin in a living organism, including organ development, disease occurrence, and drug responses. To date, AI has driven the revolutionary advances of human organoids in life science, precision medicine, and pharmaceutical science in an unprecedented way. Herein we provide a forward-looking review, the frontiers of living human organoids, covering the engineered construction strategies and multidisciplinary technologies for developing living human organoids, highlighting the cutting-edge achievements and the prospective applications of AI in organoids, living human organoids, particularly in biological study, disease occurrence, disease diagnoses and prediction, and drug screening in preclinical assay. See, this is why they're not LARPing in Congress with their DOD, or excuse me, sorry DOD, FDA Modernization Act in 2022. They really like those assays and what it allows them to do with drug discovery. Okay. Moreover, we shed light on the new research trends harnessing the power of AI for, I believe I have to keep saying this, living human organoid research in the context of multidisciplinary technologies. The aim of this paper is to motivate researchers to explore organ function throughout the human life cycle, narrow the gap between in vitro microphysiological models and the real human body, accurately predict human-related responses to external stimuli, cues, and drugs, accelerate the preclinical to clinical transformation, and ultimately enhance the health and well-being of patients. Key words: artificial intelligence, human living organoids, organoid intelligence, in vitro miniature organ models, multidisciplinary techniques, preclinical to clinical transformation. Welcome back to biomedicine and biomedical engineers and microfluidics, 3D bioprinting, wireless tissue engineering, biosensors on the bottom, data mining and analysis, optimization of culture systems. Oh, this is another one of those moments where I ask you again, can you explain to me why you wouldn't tell people about all this, given how excited you are to make sure they all know how far along you are with growing them any liver they need and just installing it into them? Not a problem. When you think people would want to know that, you're going to cure all their cancer, no one's ever going to need to be an organ donor again, etc. Sounds pretty exciting, doesn't it? The emergence of big data has promoted the training of AI algorithms on large data sets, thus improving the performance tasks. And just to throw this out here, that might be another reason why the government is not so happy to share or willing to share all these body area networks and the medical and the HIPAA associated with the electronic health records for things like what? What? Oh goodness, excuse me. Therefore, we can't help but think about how AI technology can accelerate the development of human living organoids in fundamental research and clinical therapy in biomedicine. In the context of technological innovation, researchers at Johns Hopkins University in the United States have creatively conceived a technology similar to human organs, organoid intelligence. Brain organoids as hardware. Uh-oh. I know, right? They would never, blah, 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 blah. I know. You guys just can't let go. It's okay. It's all good. We got you. Here on the channel, we've been doing the hard work for you. Yeah, we've been confronting this and going, whoa! Living brain cells wired into organoid on a chip, biocomputers, can now learn to drive robots. Hey! Thanks to open source intelligent interaction system called Metabot. This remarkable project aims to rehome human brain cells in artificial bodies. Well, let's try reading it a different way. Okay. Brain organoids as hardware to develop biocomputing. Let's see, that says 39. Hang on. Oh, there's a Google Scholar for it too. Organoid intelligence, the new frontier in biocomputing, and intelligence in a dish. Because we need that. You know, we needed that. So in 2023, this team envisioned computers running on human brain organoids. What are those again? Johns Hopkins University in the United States. I thought the Illuminati did this. I thought the Masons did this. I thought a gazillion religions did this. I thought we just didn't know. I thought that, but look, again, it's Johns Hopkins University in the United States have creatively conceived a technology similar to human organs, organoid intelligence, brain organoids as hardware to develop biocomputing. In 2023, this team envisioned computers running on human brain organoids and announced related research plans. In the rapid development of the information age, harnessing the power of AI for human living organoid research combined with gene editing Imaging analysis and multi-omics sequencing could aid in organoid system improvement, clinical decision making, and clinical trial performance analysis. AI can predict the differentiation pathway of stem cells, infer the production and development of human living organoids, and ameliorate the human living organoids culture environment, attempting to build more sophisticated and authentic in vitro physiological models in a highly human relevant manner. It's right there. So what do we got? We have PubMed, review of artificial intelligence applications and algorithms for brain organoid research. We have deep learning predicts the differentiation of kidney organoids derived from human-induced pluripotent stem cells. We have convolutional neural networks can predict retinal differentiation in retinal organoids, neuroscience, and machine learning for stem cell differentiation and proliferation classification on electrical impedance spectroscopy. Bioimpedance. Synergists now know what that means, because your body's electrical. Come on, you. There we go. There she is. February 2023 at the Frontier Scientific Journals again. Center for Alternatives to Animal Testing, Chemical and Biomolecular Engineering, Materials Science and Engineering, Comprehensive Cancer Center, Computational Sensing and Robotics, Oncology, Ophthalmology, University School of Medicine, Bioethics, Department of Biomedical Engineering. Yeah, I'm still looking for the Illuminati. Oh, hey, there's Luxembourg. Biomedicine. Oops. Recent advances in human stem cell-derived brain organoids promise to replicate critical molecular and cellular aspects of learning and memory, and possibly aspects of cognition in vitro. Coining the term organoid intelligence to encompass these developments, we present a collaborative program to implement the vision of a multidisciplinary field of organoid intelligence. Starting to see why South by Southwest was totally cool with this. Yeah, they've been working on it quite a while. Key points. Biological computing or biocomputing could be faster, more efficient, and more powerful than silicon-based computing and AI, and only require a fraction of the energy. Organoid intelligence describes an emerging multidisciplinary field working to develop biological computing using 3D cultures of human brain cells, brain organoids, and brain-machine interface technologies. Hang on, I'm getting over the shock. It's, I'm just so shocked that you be hooking this up the same exact way that, you did before and that you do, where you take somebody's neurons in a dish and then you're connecting their brain-machine interface with their digi-twin. Does anybody remember that part where we went over this stuff back in late 2023 and then into 2024? Yeah, there's a bunch of people that do, because when I found it, I was horrified. I'm like, oh, well, my goodness, North American Operators Guild for the Visible Light Communication. Then the Ariana Grande video, jumping in and out of bodies, and I tried to help people understand. I'm like, these are sciences. Right. And what are they sciences for? Regenerative medicine. Organoid intelligence requires scaling up current brain organoids, so we got to get more complex here, into complex, durable 3D structures, enriched with cells and genes associated with learning, and connecting these to next gen, here's your favorite, next generation, input and output devices, and the AI machine learning systems. Organoid intelligence requires new models, algorithms, and interface technologies to communicate with brain organoids, understand how they learn and compute, and process and store the massive amounts of data they will generate. Organoid intelligence research could also improve our understanding of brain development, learning, and memory, potentially helping to find treatments for neurological disorders such as dementia. Ensuring organoid intelligence develops in an ethically and socially responsive manner requires an embedded ethics approach, where interdisciplinary and representative teams of ethicists, researchers, and members of the public identify, discuss, and analyze ethical issues and feed these back to inform future research and work. Well, that would be great if you could admit that this is even here. It's only for us at Harvard, Sabrina. Clearly. Technologies that could enable novel biocomputing models via stimulus response training and organoid computer interfaces are in development. We envisage complex networked interfaces whereby brain organoids are connected with real-world sensors and output devices, and ultimately with each other in sensory organoids, like retinal organoids. Yeah. And are trained using biofeedback, big data warehousing, and machine learning methods. In parallel, we emphasize an embedded ethics approach to analyze the ethical aspects raised by organoid intelligence research in an iterative, collaborative manner involving all relevant stakeholders. Write your letters. Stop killing the trees. It's just an email, Sabrina. I know. Integrated microfluidic perfusion systems. Let me show you what this looks like, because you guys have seen it many, many times. Hey, could you stop painting and doing artwork, any of you, long enough to explain this to people? Because you said, this is wrong. Yeah, it's also already here in like mega in development, and a lot of people want it. So can we please talk about it for the memristor microfluidic artificial neural network way they've been using it with free space optics since the 90s? I mean, I'm glad that it took you till now to figure out that it's wrong, but can we talk about it for how it actually works? That would be great. You know, because right now the president just got done letting people know that our healthcare with electronic health records is going forward exactly the way that they're learning all about at Harvard and everywhere else. Is kind of regenerative medicine kind of important, especially with the biocyber interface, cracking the bioelectric code, and being able to offer people all these amazing new discoveries like human on a chip. I didn't say it. Classical component pathway inhibition in a human on a chip model of autoimmune demyelinating neuropathies. So when I tell you that they're under the skin and that they're very happy 3D printing and doing stuff for life extension technology, do you see what I'm scrolling through? That's your Google Scholar papers, contributor papers, yeah. Okay. Human organoids model systems for human biology and medicine. Here we go again. Excuse me. Nature reviews molecular cell biology, human organoids model systems for human biology and medicine. Here we go. 2020. The historical reliance of biological research on the use of animal models has sometimes made it challenging to address questions that are specific to the understanding of human biology and disease. But with the advent of human organoids, which are stem cell-derived 3D culture systems, it is now possible to recreate the architecture and physiology of human organs in remarkable detail. Human organoids provide unique opportunities for the study of human disease and complement animal models. Human organoids have been used to study infectious diseases, genetic disorders, and cancers through the genetic engineering of human stem cells, as well as directly when organoids are generated from patient biopsy samples. This review discusses the applications, advantages, and disadvantages of human organoids as models of development and disease, and outlines the challenges that have to be overcome for organoids to be able to substantially reduce the need for animal experiments. Yeah, we don't want to hurt the poor animals, so we'll just go ahead and continue extracting cells from humans with molecular communication. I mean, how are they going to know? They don't even believe their body's electrical. They don't have any idea what intra-body networking is. Perfect! But all the rest of you know exactly what that is. I mean, even Ariana Grande's out there making videos about jumping in and out of other people's bodies. It's just so funny. Kind of like wireless drugging. Are you laughing? They are. Mark 5:36, amen. And remember, just because so far all we've seen is a whole lot of bad stuff, that doesn't mean it has to be bad forever. The problem is all the secrecy around all this. And I'm really having a hard time believing that they're going to make this so readily available to everybody in a way that actually relieves the body autonomy problem. Because if you don't want to be hooked up to the intrabio nano things and have a bunch of nanobots running through you, you're going to have a very difficult time with this. Okay? And for those of us that have already been tested on and we don't want to be hooked to the cloud, but we know that it's already happened, etc., we're also having a difficult time with all this. So it's just something to think about. Mark 5:36, amen.